Okay, well, thank you for coming back. Looks like most people seem to want to do this remote, so might be easier to do that. But anyway, for this week, we'll be in person, and I hope it's okay. This is our fourth lecture for E340, 342. And we have, as usual, the course being live streamed and recorded. So please do remember that. So today I would like to start talking about rate laws. And so last time at the very end, we were talking about columns to output and Tellurium. I want to do a quick exercise on that, just applying those. And then I want to do a little bit of NumPy Python with HStack and VStack, which you need to be able to manipulate files. And then we will go to do some work on Michaelis-Menten rate laws and cooperative rate laws, Hill equations. And then depending on how far we get, we may talk a little bit about how they are similar to or different from the systems that they're trying to model. So to begin with, I'd ask people to do quick one slide presentation of the papers that they were looking at as possibilities. And so I was hoping that I could have people do that. And so I wondered, let me see if I have participants list. It's going to be a little tricky to do this. This would be a lot easier to do with if I were at home. So I was hoping the way we could do this would be to have people screen share their slide or two, tell us what the papers that they had submitted last week were, very briefly, and explain to us why they're interesting and what their thoughts were. And we've got quite a few people in the room, so probably we need to keep that relatively short. Next week, I'm going to be asking you to present your ideas about a project. Again, the goal of that is to team people up. I know some people had set up some online systems for doing that. I hope that's been going all right. So does anybody want to get started showing us what their paper ideas were from last week? I can just go down the list if we like. And if you're not ready to do it, that's okay. You can tell me and we can do that. So does anybody in the room here want to do it? And Sean says your PowerPoint is more about next week. That's okay too. We could get started on that as well. Anybody? Joel? Okay, why don't we I'll go through the room here first. There are 4 people in the room, and then we'll go down the list on the online, okay? So I will turn the share over to you, Joel. And I know somebody pointed out that I had forgotten to hit the publish button on the assignment to give you the opportunity to upload your ideas about your projects. So that should be okay now. I did that just an hour or so ago. You ready to present too? Okay, do you want to go first? Just again, we have a lot of people to get through, so the order, I didn't have a formal order list, so why don't you go ahead? You should be able to screen share.
So this is actually on our project.
I would unmute yourself and let me mute me. So
Okay. So, oh. Okay. So yeah, Gabe and I are doing a presentation on our project's going to be on GRN based morphogenic robots. Basically the only difference between what the literature does and what we're planning to, or the main difference is that we're going to limit cellular growth so as to focus on dynamics on networks. So that way it doesn't go outside of the scope of the class. Did you want to do the I guess talk about what we're actually going to do in it, and then
so we're basically we're going off of a main paper. I actually got that on my laptop, but really briefly, we found this paper by this guy named Peter Eggenberger, and he's evolving the paper is called Evolving Morphologies of Simulated 3D Organisms Based on Differential Gene Expression. And basically they use GRNs, and with the concentration differences for different proteins, that produces different results. So we've still got to go through the paper, and that could be something that we go on more into next week. But for right now, I think just as a general overview, this is kind of what we've been thinking about.
Yeah. So generally, actually getting into some of the specifics, I guess, is that it would basically be using evolutionary algorithms to try to train a morphogenic robot to basically take in sensory input and then output motor control. And then by using evolutionary algorithms, you can try getting the best gene regulatory networks to have the cells self-organize to move towards good stimuli and away from bad stimuli. Ideally, based off the paper that Gabe was mentioning, it would the body would also be its controller. So each cell would take up physical space, but also those cells would also be the controllers for the robot. And then the dynamics would just be that we would just plot the concentrations of proteins within the cells over time to see how the cells differentiate each other, and also to see what's the difference between successful and unsuccessful robots. But yeah, that's basically it.
So is the robot a single cell, or is the robot an ensemble of cells?
It's an ensemble.
That sounds like a 2-semester project.
Yeah, I think it's definitely gonna be. We're starting with a big foundation, and we're gonna
funnel down.
I mean, the only reason I'm saying that is that the spring semester we teach agent-based modeling, and so that would be an ideal project.
The spring semester. But of course, if you could design the control network this semester and then put it into the agents next semester, that might work. So it's possible. And you can try going all the way with it.
Yeah, originally we had wanted to do this to focus on things like regeneration in the olfactory epithelium and something like that. So, but we were starting way too. Huge.
it's
and then this has been chipped away even from that. So, but maybe by next week you'll see something that's much more specific. Hopefully, that's the.
Yeah.
But conceptually, it's very nice. I mean, there's there's sort of the things that I think about at the spring more than the fall. There was a nice series of papers about
20 years ago by Pauline Hogg.
HOG. PAL. Sorry, PALIN. I should know that well enough. Pauline Hogg.
Dr. Glazier,
But knowing Dutch is always a little problematic.
Oh, this is I think Eden's there on Zoom, and I think Eden read some of her stuff. Is that? I can't actually see him. But...
Yeah, I talked about some of that stuff in
Oh,
Eduardo's
Yeah,
class.
she was one of the people who really was critical in developing this methodology that we use in Computational. Right. These days, people tend to say cellular postmodel, but she called it GGH, Glazier Grenier Hagovec model. So she was the first person to try to do the chemotaxis component of it. Great scientist. did a lot of beautiful work on evolutionary morphogenesis. I mean, the other person who did the kind of thing you're talking about, although in a sense in a much more sophisticated way, was somebody who was here at IU, was Larry Yeager. And he wrote a software package called, boy, I'm gonna block on it now. Polyworld. And that was a bit more net logo-y in terms of the way it worked. But he had neural networks that lived inside of each of the agents. And they were quite sophisticated in terms of what they could do. I was always really impressed by that. But then he had been at Apple, and he came to IU and was on the faculty for a while, and then decided to go back corporate. So, although he still lives in Bloomington. So, a project like that, you might ping him, find him on Facebook, and ping him, and he might give you some help. He's a really nice guy. So again, I think the thing there is just because it's such a big project. If you can think about how do you design the control network to make it something practical, and if you get through that in a semester, that's plenty. And then if you do the class with me or with Randy or somebody else next semester, integrate. And again, if you can do it all in one semester, that's fine. But don't push yourself too much on that. That's great. Fascinating problem. I don't know the two papers you cite, so I need to sit down and look at those. So maybe next week you can show a few pages from those papers to show what they were able to do. That'd be good. Okay, any comments from the room? People should feel free to dive into the conversation. I know there are people with some expertise. Connor, this is your world more than mine, maybe.
I mean, I think this
Okay,
looks
so you
really,
talk
really
to him
good.
every day anyway.
Yeah, I was about to say, these are all people who work in my home lab. So
Yeah, 'cause we actually can't hear.
Wait, Connor, yeah, because it's complicated. If I keep, because my volume's off, or I guess I can just keep on the mic and the volume at the same time.
Okay, sorry, there we go. No, I've got it on. We just have to make sure there's only one turned on.
Exactly. Okay. So, Connor.
Go ahead, Connor, if you have any comment.
oh no, I was just saying that I think that like the only thing is that it seems like a lot. I'm not sure how the details will fit together, but also I think that like I was mostly saying that I don't think I can make a comment at this point, and you specifically pointed me out.
That's fine. I was just thinking it's the kind, I mean, so I was right to think it was your style a little bit.
Yeah, no, it definitely fits into the realm of interest.
Okay, great. Okay, who wants to go next? Joel, are you set up now?
Define local. Anyway, so last week I present a little bit of my virtual cornea project. But this time around, I want to show a little bit more of what exactly I'm talking about. I'm talking about a very specific part of the cornea, which is just the epithelium part of the cornea. So the cornea actually is this whole thing here, right? And most of it is stroma, but I'm focused for now for this class only on the very first layer. And placeholder. So the model that I designed was basically already done. It looks like this. It's just focusing on the green arrow. The red arrow is, as you can see, simplifying very much the other part that's called limbus, another part of the cornea. I already did some calculations here. Here is a homeostasis that I introduced. It's nothing fancy. I'm just changing the rates And here is a different approach that I'm taking. It's even good that I can test with you guys and you, James. So since the organization part of the cornea is actually 3 layers of wing cells and 3 layers of superficial cells for every columnar or basal cell, I thought of thinking about it as a column, right? Generating the cells as columns. So I found this ratio to be the differentiation ratio here and the doubling of basal cells, the thing that controls basically how much I can output with wing cells. This is an evolution since, as I mentioned before, the red space I basically turn into one cell, and now I'm introducing another cell, the transient amplifying cell. And I want to see how that changed. I already did some calculations here, but I haven't yet plotted or implemented new code. So that's, anyway, then it's introducing wound to this system and seeing if we can recover, which goes a little bit with what we read this week of stability of the system. So I'll probably do some stability analysis of this model, see. And then the spatial components, which is next week, next semester, if you guys are interested in. We have to think about some stuff about on these cells that goes a little bit beyond of just population, right? Or how they're located, what they're in contact with, and stuff like that. But that's for the future. I don't want to take more time. Maybe some people want to share more stuff.
Let's see. Any comments or questions about that? Again, some people may be, as I've said many times, people come to this class with many backgrounds and levels of preparation, different interests. And so it's perfectly fine to do something very sophisticated and very focused. And it's also perfectly fine to pick a basic paper that's educational and simple and do that. The point of the class is to learn. And so there's no rule that says that if somebody does something very elaborate that everybody does. So don't feel, you shouldn't feel ever that there's something that's too simple for the class or too sophisticated. It's really, the goal is to make it something that you feel that you learned from. All right.
Sorry, could someone unmute?
Okay, I'll speed through these pretty quickly and provide more info next time. So telomerase is an enzyme that is responsible for adding more DNA onto the ends of chromosomes when they divide. Because normally when cells replicate, they also lose a bit of the ends of the chromosomes just because the process is imperfect. But this can also cause problems in cancer because this telomerase makes them immortal and does not trigger a certain state of ending the, it's called replicative senescence, and it's when the cell stops dividing. So right now, I'm at a very general stage in my research. I've been reading a lot more of introductions rather than trying to drill down to something specific. So some questions I've come up with as I've gone along are, why do they shorten? And there's certain processes behind that. They're kind of well understood, kind of not. There are also a lot of models already out there about modeling telomere length. So creating just another one could be useful, but that being said, maybe I don't want to do something that's already been done. And for the third one, this one's the one that I'm probably most interested in. There are several drugs out there that are kind of chemotherapy drugs where they inhibit the role of telomerase in cancer. So where these cancer cells normally have a lot of the enzyme, these will help to tone it down and prevent the cells from becoming immortal. But even though those drugs will halt tumor growth, the fourth point addresses what else will happen in the body. So would that also mean that you have other problems because your telomerase is completely shut down? So investigating some of the consequences of chemotherapy would be a focus there. But to wrap it up, there are some figures that I found that have to do with cell death. So normally you want your cells to die, in fact, if certain things go wrong, but if it's cancerous and it does not have that particular gene called P53, then they'll continue living. So these graphs model what happens when a cancer cell has particular attributes that allow it to perpetuate its existence. So to connect it back to this course, I could possibly go down the route of a more protein signaling pathway because this is an enzyme, or I could go into the metabolic network side because these particular drugs need to metabolize somehow. So that's all.
So there are a lot of possibilities there. Just turn on and off is something that is potentially interesting in its own right. In a sense that some of the things that you're talking about are inside it's the kind of questions that Joel was asking just a second ago. Because you're asking a question about the sort of regulation of survival of single cell, and he's asking more a question about tissue regulation, which is you want to know how you keep homeostatic mechanisms. There's always this conflict in our bodies in soma, which is you need some stem cells because you need to be able to replace damaged tissue. But stem cells intrinsically are cancerous. And so you need to be able to keep them under control. And so Joel's talking about essentially stem cell niche maintenance. How do you have a tissue be able to regenerate at the same time not produce an excessive number of stem cells or lose them? You have a little bit of the same issue within an individual cell. You want to be able to replicate. If it's stem-like, it should be able to replicate as many times as it needs to. On the other hand, once it becomes somatic, you don't want it to be able to replicate an infinite amount of times because then you have the problems you mentioned with cancer. And this relationship to P53, of course, is an interesting one. As you've heard me say before, the only gene that is affected in most cancers is P53. People think of cancer as a genetic disease, but in fact, there are a couple of genes that are relatively commonly affected in cancer: APC, RAS, RHO. But beyond those two or three, there is no gene which is mutated in more than about 1% of cancers. No.
Something goes wrong.
No. So there's this famous study of Carl O'Malley, which asked the question. So there's this beautiful study. There was this beautiful study from Hopkins a few years ago, which showed that effectively the probability of a tissue developing cancer is linearly proportional to the number of stem cell divisions. I mean, it's not perfect. There always are prefactors, but in a sense, the probability of a tissue developing cancer is linearly proportional to the number of stem cell divisions. And what that means is that if you're a whale or an elephant, you would expect to get cancer right away because you've got a very large body, and so you have a lot of stem cell divisions going. And so the question is, why don't elephants get cancer? They live a long time, and they don't get cancer. There's some large animals like horses that actually do get cancer fairly easily. So it's not universal. And one of the ways that elephants deal with this is that they have lots of copies of P53. And so the experiment that I really wanted to do, which I never was able to do, would have been to engineer mice with duplicated P53. Because mice have a short life expectancy. They develop cancers fairly early. And so if you could duplicate P53, you might be able to suppress cancer in mice. If it worked in mice, it should work in humans. Well, because they don't need to, right? I mean, you don't need to do all of the ROS buffering, since if you're only living 2 years, you don't have to worry about that so much. But it's an interesting question. You could say, well, P53 you think of as an apoptosis gene, it's a cell death promoter. And so if you doubled it, you would expect terrible things might happen. But if it's tightly regulated, the number of copies of the gene won't change the amount of protein. And so it's certainly in the case of elephants, they don't have more P53 floating around. They just have backup copies so that if something happens to one copy, they've got additional copies that can express the function. There's also something called P63, which is not something anybody talks about, which apparently can work as a backup for P53 in humans, can be turned on. It's been investigated as an anti-cancer treatment. So that's great. Again, I think these are all great topics. And I made the comment about the difference between a topic and a project. And so now the key thing will be trying to take that wonderful idea and narrow it down to something that's achievable in the cement. Lots of great problems. Okay, who would like to go next? Let's see. Eden.
Perfect, because Connor and I are gonna go together, so we were about to synchronize. Do you want to share or do you want me to share?
You can go ahead and share.
Yeah. Let me find it. Okay. Everyone can see that, yeah. So
I want to hit slideshow so it's bigger for them.
Where?
Up top right.
Thank you.
Yep.
Okay. So Connor and I were originally thinking of our own projects, but then we were talking the last couple of days and are thinking of converging based on sort of similar methods and a similar underlying question. So I was originally looking at biodiversity and the emergence of niches as a catalytic process. And I was interested in modeling that looking into things like autocatalytic sets and things like that, trying to find network approaches to it. And so I was reading papers to that end, and then Connor can speak for himself.
So kind. Yeah, so on the flip side of that, I was more interested in the kind of circular organization of chemical species. There's this theory in cognitive science called autopoiesis, which is pretty strongly connected to ideas involving autocatalytic sets. And on top of that, there's connections to these ideas in viability theory from like earlier cybernetics that has started to make a comeback. But there's additional nuances and issues when you're having to consider a system and its self-maintained boundary as opposed to just a network. So Eden and I are interested in kind of existing at a middle point where we just look at organization of species, not worried about boundary, and try to take on a graph theoretic and dynamic approach that could be applied to ecological logic as well as chemical logic.
Well, it's not maybe the best one to do as a antimony tellurium model, but I always thought a nice simulation to do. It's a simple one, although I've never run it, so I don't know if it actually works. Would be to do the periodicity of cicadas. And so you have one parameter, which is the year period of the cicadas. You have a small mutation rate. If you cross a 15 with a 13, you get a 14, for example, at least with some probability. And then you have a predation factor that's sharply saturated, so that if you get lots of individuals in a year, then your survival rate goes up faster than linearly.
Mhm.
And that should, if you start out with more or less random periodicities in your organism, it should give you the relatively prime 13, 17-year cycles. I'm sure people have done that. And I haven't. I should probably Google it and find it in a minute. But that's always struck me as an example of the kind of sort of simple evolution where you, because normally one thinks of the Darwinian evolution, you have to have some kind of boundary. Darwin always thought of spatial boundaries, or it could be niche boundaries. But in this case, you have basically a viability boundary, which is hybrids between year A and year B are going to come out in a year when you have fewer individuals, and therefore they're going to have lower survival. The probability of getting crosses will be higher if you have two broods with different numbers in the same year. And so the way to minimize the probability of clobbering yourself with another brood is to have relatively prime numbers that are fairly big. And so at least theoretically that ought to work. And as I say, I'm sure people have done it. Again, to me, that seems like fundamentally an agent-based model, but that's fine. But that could be possibly one to think about, where you're trying to think about something that's speciation that's both synchronous and sympatric is hard. If you have spatial division, then it's easier to get speciation. But to try to actually get speciation within a population that can interbreed at the same time and is co-located takes a little bit more work. I always think for that kind of modeling, more on the autocatalytic side, Chris Adami at Michigan State wrote a little autocatalytic network simulator called Avida. It was like many of these sort of Farmer and company autocatalytic models. It was a little bit artificial in the sense that what you have are your strings are in fact sequences of instructions, computer instructions. And you have to, it's a little bit of a cheat, because if you have a copy instruction, then you could have everything work. But to try to evolve a copy without putting it in by hand, boy, it's hard. I mean, as Holland likes to say, you get a lot of mud when you do those kind of simulations. So good. Well, I think people are doing well. I think between now and next week, you can keep refining these ideas. And it's great. Okay. Okay, so who's next? You've
I can go
heard
next.
Sabine, Connor, Sean. Please go ahead.
All right, sorry, just making sure I can go off mute here. You guys should be able to see PowerPoint. Is that correct? All right.
That looks great.
So yeah, I'm going to try to attempt to model some AMPA receptor trafficking for hippocampal neurons in juvenile and adult rats. Kind of just to highlight what that even means. Last semester, I took a neuroscience of learning and memory course, and in it they talked about this long-term potentiation, or LTP, and long-term depression, LTD, as this potential mechanism for learning and memory. It's measurable through the electrical potentiation. You can see a graph here on the bottom right that kind of shows what a graph like that would look like. But one of the key features of LTP, LTD is that AMPA receptors and NMDA receptors get trafficked to the synapse, which kind of this area here that my mouse pointer is, or sorry, this area here that my mouse pointer is on. And, you know, this trafficking occurs where AMPA receptors can live in this endosome where they're recycled, turned over, you know, for protein turnover, but the different subunits could exist here. They can be trafficked out to the external cell, but not necessarily at the synapse, and then they can be trafficked to the synapse. So as you might be able to guess, it's kind of what these models focus on is tracking these AMPA receptors between the various locations. And the model that I'm going to be starting from actually uses the AMPA receptor count at the synapse as a proxy for determining whether LTP or LTD is occurring. These models focus on the adult rat hippocampal cells, and specifically these adult AMPA receptors are made out of different subunits than the subunits that make up juvenile AMPA receptors. And one of the key features that the literature talks through is just the difference between these. And so I'm hoping to kind of get at that with this. So this is a picture of the current adult model. In it, calcium is the main influx, it's the main input. With it, you can, if you have a steady influx of calcium, you'll see just a kind of steady line here of AMPA receptors at the synapse, which is now what this is measuring. But then if you were to spike the influx of calcium, you would see this increase of AMPA receptors trafficked to the synapse. And then the converse for LTD, where they're moved away from the synapse. And so in terms of what is known, as I just said, there's this trafficking model that's specific to adults. It's backed by the literature, different knockout experiments, pretty well-known, robust model, but they don't really bring in the subunits. They don't really hit at these juvenile AMPA receptors. They just kind of like chalk it up, just saying like, oh yeah, it's not really relevant. But the literature clearly says that there's these a 4 to 5-fold difference in terms of the efficiency of these AMPA receptors, namely that the juvenile AMPA receptors are much more efficient at firing from calcium than the adult AMPA receptors. And pretty early in the rat's development, I think it's 3 weeks, almost all of these juvenile AMPA receptors have been turned over into the adult AMPA receptors. And so I think that's kind of interesting, like why are the less efficient AMPA receptors the go forward, the status quo in the adult? Like in my mind, it would make more sense to use the more efficient ones. So anyways, I have that starting model of the adult AMPA receptors trafficking. The literature says there's this difference. I have found a molecular model using like kinematic equations of how the subunits for both juvenile and adult AMPA receptors are created. So I'm kind of hoping to incorporate those ideas into the model so that I can like kind of set which AMPA receptors, either the juveniles or the adult ones, I'm utilizing. And so my hypothesis is that I should be able to put have both of these AMPA receptors in a single model. And with that one model, I should be able to show that LTP occurs as it did in the original model with, let's say, 5 units of calcium influx to produce the LTP. And then I'm hoping to also be able to show with a 5-fold decrease that I can get these the same LTP to occur as if it's like this juvenile AMPA receptor. And so this was just kind of answering some of the questions that were asked in terms of the input and output, but I'm measuring the same things that original graph was measuring. And then the NanoHub project, just kind of showing the actual networks and showing the LTP and LTD occur. It'd be interesting if it doesn't work, if I'm not able to reproduce the juvenile side, if there's time to try to perturb the network around those juvenile AMPA receptors and see if maybe there's like a clear inhibition, or, you know, just seeing if there is something there that could lend to the biology of like a next steps of where to look for what to be included into a next iteration of the model. But that's it.
Well, you've got at least 3 very profound biological phenomena that you're thinking about here. One of which is trafficking and recycling of receptors. When the receptor is activated, it's endocytosed, it's reprocessed, and then returned to the membrane. Another one is, actually I've got to come up with 4 rather than 3. That's the first one. The second one is the switching between juvenile and adult forms of the molecule. And this happens a lot, and it's often not exactly clear why it happens. If you look in humans, there are dozens of places where there's a juvenile form of a protein and an adult form. Sometimes there are multiple juvenile and multiple adult forms, and some of them persist and some don't. And in fact, in the disease that we've worked on with the School of Medicine, polycystic kidney disease, specifically, there's a juvenile cadherin in the kidney that doesn't get turned off in some cells. And the expression of that juvenile cadherin in adults is sufficient and necessary to trigger the disease. So in that case, you have to turn off the juvenile form, probably because the juvenile form is associated with the growth of the kidney. And so if you don't turn it off, the kidney doesn't stop growing properly when you become an adult, because there's a big mystery about how the organs stop growing. In particular, how to not only how do organs stop growing, which is already a problem, but how do you get symmetry between the left hand and right hand side of the body when you don't have any communication between them? Right? Your legs are approximately the same length, but there's no communication during growth between them. And so you need to actually have pretty accurate mechanisms for shutting things down. And so that switching between juvenile and adult forms, things like hemoglobin switch, which are sort of surprise, as well as signaling a molecule. So that's a fundamental one. The next one that you've identified is the fact that a lot of these receptors are polymers. They're well, not polymers, but are complexes where they're composed of multiple subunits. And it's not always clear why you need the multiple subunits. In other words, if you look at, say, growth factor receptors, FGF receptor 1, FGF receptor 2, FGF receptor 3, they'll complex to do, in that case, dimers or pairs of dimers.
Mm-hmm.
And it's not exactly clear why you need to have, clearly you can have different tune the response to the signal that way, but it's not exactly clear what it's doing. If you look at models of this, the models work pretty well without having all of that additional complexity of having multimeric receptors. And so that's something you were talking about. And then the last one is the role of calcium. And one of the reasons I became interested in biology in the first place was many years ago, having resisted doing any biology for my whole life, well, up to that point, I was reading Darnell Lodish Baltimore Introductory Biology textbook, Introduction to Cell Biology. And you read something and it says, there's an influx of calcium into the cell and the cell does this. And then you read 2 pages later, there's an influx of calcium into the cell and the cell does something totally different. And then 3 pages later, you read something and says, there's an influx of calcium into the cell and the cell does yet another thing. And at that time, there was essentially no discussion of how the same calcium signal led to very, very different specific cellular responses. Now, at this point, I think a little bit more is known about that. But there's still a lot of ambiguity about how that calcium signal leads to specific response, because calcium influx is a trigger for so many cell activities. How the cell knows which one is the right one is still a bit mysterious. People like Hoffman talk about temporal coding as one way. Is the calcium signal periodic or not periodic? Is it slow? Is it fast? How big is it? And that certainly may be part of it, but exactly how that works is not so clear. And so, and this is also a nice model in the sense that you have a signaling and gene regulatory network model, and you also have a transport model, right? A is essentially a classic regulatory or signaling network, mainly signaling in this case. B is a transport model, right? Because your ampere is being moved from endosome to exosome and so on. And so you have a more of a PBPK model in B and more of a classic dynamic network model in A. And so you've got both kinds of approach, compartmental modeling and molecular dynamic model. Great. Okay, who's next? I'm sorry, I don't have a piece of paper here to keep track of who we've gone through.
Professor, I'll go ahead with my
Okay,
presentation.
please go ahead.
Mm. Hello everyone. So we are a team of 3, and me, Dhanusha, and Purva will be working on the computational model of EGFR and ERK pathways in cancer. So everyone knows. Okay, so let me elaborate. The EGFR is this pathway is one of the important pathways in human actually. So what it does is it's regulate the growth of cells in few of regulate the cell growth. So our first approach of getting to know about cancer was to know how the cell growth is regulated in human body, so that if any cancer or any kind of tumor is happening, can we regulate it, can we restrict it, or can we just eradicate it completely through surgery or anything. So our approach was to study the pathway so that we can frame a pipeline to how to treat the cancer cell. So this was our first approach. So now going to the next slide, EGFR is the epidermal growth factor receptor, which belongs to the tyrosine kinase superfamily. There are a lot of papers which are working on the ERK and EGFR relation between the EGFR and ERK pathways. So our first approach was to get to know the EGFR and the relation with phosphate. So in case of EGFR, what it happens is when EGF is converted to EGFR, there is a autophosphorylation. So there is an increase in phosphates in the surrounding of the cancer cell or in the human vasculature. So the first point we should, our focus was to regulate the phosphate so that either it will restrict the cancer growth, or it hampers the healthy cells around the cancer cell. So next slide says the basics of EGFR and ERK pathway. So this is a schematic of the EGF pathway, starting with the EGF binding to the EGFR and ending with the ERK complex. This complex generally creates this compound, the combination of EGFR and ERK creates, helps the cancer cell to grow indefinitely without having a restriction of the cell. Like everybody knows, there is a cell cycle of G0, G1, G2, and then there is senescence of cell. But in case of cancer, there is no stop. There is no traffic signal, I would say. So to restrict, to have a traffic signal to stop the flow of the cancer cell or the growth of the cancer cell, we need something that should restrict the cancer growth. So that's where EGR and ERK comes into place. This is a very complex. In this slide, I'm presenting the pathway of EGFR and ERK. This is a very complex part. So rather than going deeply into like modeling the whole network, our focus would be to implement the relation with phosphate with EGF rather than completely modeling the network. So for preliminary model, what we have, like, we are still looking into a few of the papers that can give us the parameter values for the equations we need, for the kinetic equations we need to model the EGFR model, and the state variables, like from where the flow is, like what if a cancer who is what if a patient is already detected with second level or a third level of cancer, what are the dietary restraints he or she should have, like should be a phosphate there should be a lot of phosphate intake or should be restriction of phosphate intake. So these are the models which we are still going through to implement so that there will be a relation between EGFR and phosphate. And so we have planned accordingly that in first week of from today and the first week will be going through all the parameters and all the literature which we can find to create the model, and subsequently will once we get all the parameters and ODs defined for the model creation, we'll build the model and get a preliminary result. If this doesn't go well, we'll go back to the EGFR and reproduce the existing existing paper which we have read for this proposal. So that's all from my side.
Okay, can we go back to the picture, your pathway picture for a sec?
Yeah, sure.
The one before that, actually.
Okay.
The receptor picture. So I just wanted to comment, here you have a dimerized receptor, right? We were talking about this, right? And they're different EGFRs. So in principle, you could have multiple EGFR components.
So in this case, we are focusing on the version 3, the variant 3, EGFR variant 3.
So but it was just interesting that somebody had brought up this issue of the multimeric receptor. I thought you had a nice example here. And then I'd mentioned, right, in the context of mutations, RAS is one of the few genes that gets mutated pretty commonly in cancer. So you've got RAS there in your picture. This is a nice, the other thing which would be nice to talk about in this class, and I've hoped over the years to do a lecture on, and somehow we get to the end of the semester and we never get to it, which is regulation of sensitivity. And so in the example that you have here, epithelial growth factor, EGF, could be like VEGF, or there are many growth factors. That can function as a survival factor or as a pro-proliferative factor. But typically, what happens in talking about, I mean, and this is coming back actually to what Joel was showing earlier in a very abstract way, where he had cells that were proliferating and sending signals that were either promoting or inhibiting the replication or survival of other cells. One of those signals very commonly would be EGF or something similar to EGF. And so if for a tissue to remain homeostatic, you need enough of the growth factor so that the cells don't die off, in which case you have degeneration of the tissue, but not so much of the growth factor that you have proliferation of the cells that destroys the structure of the tissue. And so there are two ways that can be done. One is to regulate the amount of growth factor that's present. The other one is to regulate how much the cells respond to a given amount of growth factor. And cells are pretty good at determining what the typical level of molecule in their environment is and adjusting their sensitivity to that signal in response to that. One of the ways they can do that, I mentioned as a classic example, is your adjustment of your photoreceptors to light intensity, where the rods and the cones in your eye synthesize additional photoreceptors or destroy photoreceptors. If you're in bright light, they destroy photoreceptors. If you're in dark, you synthesize more. That seems like a very inefficient way to change your light sensitivity, because you're actually having to do an enormous amount of molecular synthesis and degradation. But that's why dark adaptation takes time. It's because your cells actually have to synthesize photoreceptors. In this case, you don't want the cell needs to know if there's a given level of EGF in the environment, it has to adjust its density of EGFR or the responsiveness inside of the cell to EGFR binding so that the cells survive and don't proliferate for the normal level. There are a couple of ways that can be done. One would be to change downstream, to change essentially the amplification. Once you've received the EGFR signal, change the level of amplification of the signal downstream through the MEC and ERK and RAS. So that's one way of changing your sensitivity. The other way would be simply changing the number of EGFR receptors. You have more receptors, then you have a bigger signal for a given level of EGF in the environment. And the third one would be to change those EGFRs out, change the ratio of EGFR1 to EGFR3, and that can change the sensitivity as well. On the other hand, you don't want the cells to adapt too well, because in that case, the EGF level doesn't do anything. And so one of the things that happens classically with the classic example is breast cancer, where normally you need estrogen for the breast tissue to survive. That's great from a cancer point of view, I mean, from protection against cancer point of view, because if you block estrogen, then you kill the cancer cells. But what happens is the cells then constitutively activate the signal that's downstream of the estrogen receptor. First, they upregulate the sensitivity to estrogen. So a given level of estrogen has more pro-proliferative effect. That would be effectively increasing EGFR receptor density, although estrogen receptor doesn't work like that because it's not a membrane receptor. And then the second thing would be to just turn things on downstream. So if you turned on ERK in this diagram that you have here, independently of whether EGFR were bound or not, then you have a problem, because now you don't have any way of turning off the cell cycle. And so there actually are multiple places here where this system can be controlled or fail for homeostasis. And so Joel's model was this very high level view that there is some signal that's either pro-proliferative or anti-proliferative, pro-survival or anti-survival. And we heard a little bit more about that in the context of P53. In your case, you're drilling into how that signal works, and in particular, how the sensitivity of that signal can be regulated. And I realize I'm jumping into some pretty, compared to the lectures I've been giving, these are pretty sophisticated concepts, but you're bringing up pretty sophisticated problems. I hope that's okay. So I think there are a whole bunch of really interesting questions you could ask here. For a given level of EGF, what controls the level of ERK activity? If you change the density of EGFR, what happens? If you change the strength of the signaling through RAS, what happens? If you change the identity of those dimers at the surface, what happens? That already is a critical problem. And one of the reasons you get the cancer evolves around these estrogen blockers, for example, is precisely because there's always some variability in the way these cells respond. You block estrogen. I mean, I'm saying estrogen because I'm thinking breast cancer, but in this case, you'd block EGF, and and that you'd say, great, I block EGF, now my cancer won't grow because there's the cells that depend on it won't be able to proliferate. And the first thing that will happen is cells with a higher level of EGFR will survive. So you're selecting for the ones that avoid the drug. And then you get unlucky at some point, and some cell constitutively activates what's downstream of EGFR, whether the EGFR receptor is activated or not. And at that point, the drug no longer works. And so one of the things that people like our Moffitt friends do now in cancer treatment, that's interesting. I just saw somebody, some Facebook friend of a friend who was going to Moffitt for cancer treatment specifically for this issue, is that instead of giving the drug these growth factor blockers in the classical way, you give them at a low enough level that they slow down the proliferation, but not so much that you kill off the cells that are sensitive. And so it's possible then you don't get rid of the tumor completely, but you can keep the tumor sensitive to the drug for a longer period of time, at least maybe long enough that you die of something else before you die of the cancer. And so a couple of these talks are addressing this very fundamental question of somatic evolution, and in particular, how the system evolves. Right, our goal as engineers is to control, or medical designers, is to control systems, biological systems. But these biological systems have a lot of feedback in them to begin with, and they have this annoying tendency to evolve around the control that we try to apply to. So I think there's a lot of really great science in this one too. It ties in very well with some of the other talks. Again, I think the key is going to be trying to focus on a particular problem that's achievable. So I'd start with a published paper which you can replicate. And I really do recommend that for everybody as a beginning, so that you know you have something in hand. I understand the ambition of wanting to do something really new, but I strongly encourage people to start with a paper which you can replicate that's simple, and get that under your belt before you get too ambitious. Each one of these, I hate to say it, could be a good PhD project. And so if you all do PhD projects based on the things you do in this class, I would be delighted. But I don't, I'd prefer you not to have to wait 5 years to finish. The other thing that came up, which I didn't mention at the time, was in the neuronal potentiation, the LTP potentiation. Some of the signaling molecules that are involved in LTP were ones that we've seen in these other diagrams as well. And so another theme that we run into, and I'm trying to elucidate sort of the classic biological themes as we talk about these things, is the fact that the same molecule will participate in apparently very different processes. You'll see this happen, for example, vascular endothelial growth factor, which we think of as critical in development in cancer, also works in immune response. TGF beta, tumor growth factor beta, which is a strangely named molecule because in fact, it's primarily a molecule that's essential in developmental processes. But it was discovered in tumors, that's how it got the name, is another one where you'll see these same molecules show up again and again. Those are signaling molecules on the outside like EGF, but your SOS, RAS, and ERK are molecules that will show up all over. And we saw in one of the cancer molecule discussions, JAK and STAT. And JAK STAT play a role in cell survival and cell death. And so if you were studying immune response to infection, one of the primary ways that cells develop resistance to viral infection is using JAK STAT as a signaling pathway. And so again, just the way we talked about how calcium seems to show up again and again doing different things. You'll see things like JAK-STAT or RAS show up again and again doing different things. ERK is another one. Joel, go ahead. Why don't
Right. I guess to your point, that's why I prefer a top-down approach than a bottom-up approach, because the bottom-up is so easy for you to get lost on all the functions one molecule can have. It's better for you to start with trying to mimic the behavior, and then you look for the minimal thing you need to reproduce that effect that you're looking for. Because otherwise you can just get into a habit or rabbit hole and get lost in the literature, because the biology is crazy, the amount of stuff and the complexity is crazy.
Yeah.
So I think that that's a reasonable point of view. I would say that there isn't a right or a wrong on this. I think that it's fascinating to go from the top down and ask questions like the Komarova paper, which is, what are all the possible control circuits that lead to homeostasis of a stem cell regulation? And then mathematically, as a pure mathematician, ask, what are the mathematical control structures that are possible? And of the 64 possible structures, only 2 of them actually could work. That's pretty neat that you can do something like that. I think there's also a lot of value in looking at a particular circuit like this one, or some of the other ones we've seen, and asking the question, this is what the cells do, how does it work? And ultimately, of course, you'd like to be able to put them together, and sometimes you do get into these enormous messes of hairballs of signaling. But I think that I don't want to discourage people from taking either approach, or a middle out approach either. It's certainly difficult to try to connect these micro approaches to the macro approach. And I think one of the challenges in this field, and one of the opportunities going forward in the future, will be to try to make those connections in a more systematic way. All right. Who did we miss? Lindsay and Zachary?
I think
I'm
Zach
sorry.
went. So
I'm sorry about that. It's not good to say, but I do say, if I met somebody who did not have a name, I would remember that. Several people in the room I do recognize because I've seen them in other contexts. Many of you I've communicated with by email, but I don't necessarily always recognize people. And I did take my mask off today, but I normally wear it, and I know recognizing people under masks is also tricky. Again, at least with Zoom, if I can see the list of people, I know who's speaking. But the way I have the Zoom configured, I don't get to see that. Okay, go ahead, please.
Okay, so basically for my project, I am doing an extension of my PhD research project in a direction that I might not normally have been able to take it. I work mostly at sort of a neural circuit level. And for this, I'm looking to dig down more finely grained into a molecular level. So basically, neurons have a lot of different types of ion conductances. These are some examples around this ring here, and a lot of them are redundant. Like there's 3 calcium channels depicted here, like 2 or 3 sodium channels. So there's a lot of redundancy here. And what that means is that a single neuron can actually instantiate the same behavioral profile in a number of different ways using combinations of all of these ion channels. So if you go into the biology, and this is primarily studied, I think, in the stomatogastric ganglion of crabs. But if you go into a crab stomatogastric ganglion, pull out a neuron that is behaving in a typical way, you will see quite a range of ways that it actually instantiates that behavior. But the actual ion concentrations also, by the same token, follow sort of a logical organization. They'll often be correlated to each other. And this is what that's depicting, is just all the different correlations between ion channel species. And that's what this is over here. So if we imagine that this is a plot of fitness, and say these are like two different excitatory channels, if you want more of one channel, you have to have less of the other, and things like that can be observed. It's not advancing. There we go.
And what is tetrodotoxin doing in our world here?
That's one of the issues with the explanations that have been come up with. So that's what I'm going to get into here. So there's a lot of explanations for how a biological system might exploit this property. So basically, the thought is that if you know about these correlations, I say no in a very loose way, but you should be able to arrange your ion channel concentrations in such a way that you know you will be doing what you're supposed to be doing. And so there's a variety of ways that this could happen. One of them is called activity-dependent homeostatic plasticity, which basically is a way of sensing the calcium concentration in the cell. And then if it's too high, you might activate this gene, sort of a gene regulatory cascade, to downregulate excitatory conductances and upregulate inhibitory conductances, or something like that. So that's one possibility for how that might work. But a big problem with that is that we observe all these nice correlations when the activity level of the neuron is where it's supposed to be. But when you introduce tetrodotoxin and artificially silence the neuron, that's when homeostatic plasticity should be like the most active. It should be really trying to get that activity back. But actually what we see is those correlations disappear or change significantly in that case. So in order to explain that, people have proposed all sorts of things, like maybe neuromodulation is involved, maybe it's cell type specific. But I think that it's nothing, or basically that it's nothing more than a blind homeostatic activity regulation. Basically, instead of saying, oh, we know that these correlations have to exist, how can I maintain them? In order to be a good neuron, the system is saying, I know what a good neuron is, let's get there. And then just by virtue of being there, you'll find yourself on ridges of fitness, which have correlations among them. So yeah, I just want to show this by hooking up a Morris-Lacar neuron model to some sort of a calcium-dependent homeostatic plasticity mechanism. Lots of similar things have been modeled. I might have to simplify one, but I want to sort of build in various correlations in the expression levels of the different ion channels, and then show that it's not actually so much these correlations that I've built in, which determines what I get back out, but actually the structure of the fitness landscape.
So when you say homeostatic model, so you're thinking about basically a gene regulatory model where you have a signal that basically autocatalytic signal, or a self-regulating signal that.
Yeah, it's a lot of the time it's modeled as you using the calcium concentration as a proxy for activity.
Right.
And then that just initiates a gene regulatory cascade. Sometimes it's modeled as just embedding receptors that are already made into the membrane as a function of calcium, but I think it would be easier to do it this way.
I think it's interesting because it's hard to frame these things in a non-teleological way, right? It's easy to say the cell wants to maintain some characteristic, but in fact the cell doesn't know what it wants. It's got to be right. The homeostasis has to be an outcome of a particular network structure, and certainly an interesting question about how robust that structure is to perturbation. What's the range over which you recover How stable is it to structural perturbations? These kinds of questions. But again, I thought, like, coming back to Joel's example from Natalia's paper, right, there were 64 possible regulatory circuits that all led to regulation of transient amplifying cells and differentiated cells, and give you the right number. But only 2 of them were stable to particular perturbations of the system. That were stable in the sense that they recovered to not for finite amplitude perturbations. And so I think, again, that's a theme that there's this shared theme that keeps coming up, which is how do homeostatic networks work? How does this homeostasis actually function in practice? Whether it's implemented at the multicellular level or within an individual cell. I think that's pretty interesting. Again, I'd love to sit down with groups individually and look at some of the source papers where there's existing modeling, see how it was done and what can be done. I appreciate people's patience that I was away last week teaching, and I got back late last night, so if I seem a little bit tired today, I'm a bit jet lagged today. But these are great. Is that everybody? Have we heard everybody? Did I miss somebody?
I could talk about the paper that I looked at and what I'm theorizing.
Oh,
and
I'm
working
sorry,
on.
Grant. I'm sorry. Thank you. Go ahead, please. I didn't mean to exclude anybody, but I wasn't. Sometimes I feel a little bit like a tour guide. I'm going 1, 2, 3, 4, 5, 6. Have I gotten through everybody? But I wasn't keeping count today. So go ahead.
All right, can you guys see the presentation? Okay, so basically, for my idea for a project, I looked at a few different papers and I saw this paper, and I think that right now I want to kind of build off of / modify what this paper was working on. So this is a paper from Breakspear, Terry, and Friston, and it has this long-winded name: modulation of an excitatory synaptic coupling facilitates synchronization and complex dynamics in a biophysical model of neurodynamics. And so what the paper talks about is how they went about modeling the behavior of slightly larger, more complex neural networks inside of actual brains. And what they did for this model is they have these smaller clusters called columns of neurons that are short range, and they have like more strong connections than these what are called mesoscopic arrays, where these small columns are connected by weaker, longer range neurons through excitatory to excitatory connections. And so what they did was they came up with differential equations to model how the mean membrane potential for the excitatory and inhibitory neurons behaved over time inside of these columns, as well as the fraction of open potassium channels. And then they also came up with the same thing for the mesoscopic arrays, so on that larger scale. And so what they wanted to find was, how do these columns behave when they are independent of each other versus when they are connected? And they look at the behavior that develops once they become connected. So when identical columns are connected, that is for all these parameters that they defined that are some of them are physiologically measurable, such as threshold potentials and Nernst potentials, and then others are like functional synaptic strengths and maximum fire rates. And so that was used to define these differential equations. But so when all these parameters are the same for the two different columns of neurons, what they have when they're connected is called the symmetry manifolds, what they called them. And so what they found was when these were connected, there's they can be stable against noise and perturbations, but not for all parameters. And so what they can have is certain parameters lead to blowout bifurcation where there's a change in the periodicity. And so this is a graph of the different periodicities, and this is a graph of all the different variables of the differential equations. And so the thing is that about the study is that it goes on to define more complex behaviors, and due to like how much I know about this field, I wanted to focus in on like the first part of the paper essentially, which is the synchronization that they were measuring when they adjusted the connectivity of the columns of neurons. So for potential modifications to the study, when I focus in, one thing that they suggested doing was doing large differences in the parameters between connected columns. And so one thing I was thinking is interactions between different parts of the brain that have different like concentrations of sodium, etc., all these parameters that they use that are physiological, seeing how these columns of neurons interact between different regions of the brain, and how these different physiological differences lead to different levels of synchronization. And the reason for that is because of the idea of coherence, where highly coordinated cycles are thought to have higher communication of between neurons. So when they're firing together, they're communicating more at the same time. Or I could try to do modification to the ODEs that they came up with. They had some complex modeling for different physiological effects. So I could try adding more on top of that, or I could try removing them and seeing how that affects the chaotic synchronization that they saw between neuronal columns. And so that's what I've thought of so far.
Oh, that's great. I'm smiling for a number of reasons. One, that was the topic I wanted to work on when I started as an assistant professor 30 years ago. I never did. Well, I did a little bit, but not a lot. I did have one student who did a PhD on this topic of synchronization as a function of connectivity in cortical columns. Sridhar. Then I wound up doing more classic developmental biology. So this is a topic of great sentimental value to me. Something else that came up that was also in the previous talk a little bit, which is to me one of the great mysteries, is how neuromodulation works. Because if you take a neural network, an artificial neural network, and you say, okay, I'm going to reduce the value of my connectivity of each connection by 20%, the network stops working. And so how do you get non-specific modulation to actually have functional value in a network? Is a bit mysterious. So that's something that's of interest. The modulation of connectivity is fascinating and a great problem. Synchronization is a great problem. The other thing that I was going to say, and is that, and this was something that I wanted to add as an option, maybe it was there, And the way people are going, I don't think it would have been one that people would have picked up particularly. Somebody's working on neuromodulation. Yes, how does it work? How come it doesn't just make a mess? Computation and artificial neural networks, it makes a mess.
Right. So part of what we're adding into the artificial neural networks is the ability to choose among all of those channel types that I had on my slide earlier, which each react differently to the neuromodulation. So in that way, you can sort of tune your reaction to the neuromodulation to be in line with the degeneracy of the space that I also had on my slide. So.
That's a fascinating problem. I'm glad that you're working on it. I'd love to see those results. The other thing that I wanted to say about the synchronization problem is that, and I'm embarrassed, I'm blocking on his name. So 2 years ago, I have to check. 2 years ago, I think, one of the students in this class was a master's student who now is a PhD student at Purdue, did a project on synchronization of cortical columns based on a slightly different paper from the one that you proposed. And one of the problems that we have in this class is that people do these projects on NanoHub, and then they go and do other things usually. And sometimes the projects are fantastic, and what you get at the end of your semester is something that people will use a lot, and they're finished. But more often you have something that's enough for the project, but isn't something that is perfect. And so something that I've encouraged people to do over the years, mainly for people who didn't have an idea of exactly what they wanted to do, was to go back to a previous year project and make it better. And so it might be worth looking at, and I'm sorry, this is my jet lag talking. I will find his name and find the project. You might want to look at what he did in terms of his neural synchronization project. And I'm not saying you should necessarily take his project and make it better, but you might want to look at it because you might learn from it, not so much about the science of what you're doing, which is somewhat different, but at least in terms of the presentation, because you could say, this is how he presented the problem. Does it make sense to you? Did you learn from it? How could you present your problem better? And so that the lack of continuity, in a sense, is a problem, because to do good educational software takes a lot of rounds of revision, and we don't have the time in one semester to do that. Of course, if it's a project that you come back to because it's part of your PhD work, then you do have the opportunity to revisit it. But it's not a terrible thing to look back at previous work and say, is there somebody else's project that I could use leverage here? These are all open source projects, so you could take it and use it as you want. Of course, it's nicer if you do it with the person. You contact the person who did the original project. They're usually very happy to have somebody pick up what they did and work on that. But the neural synchronization is an interesting one. There's a lot of richness there. And I certainly remember when that first really got to be an interesting problem. I remember Kaneko, Tokyo, goes back a long way, because it's these issues of synchronization. They're pioneers in the field of computational mathematical biology, like Glass, in the context of heart modeling and synchronization. But Kaneko did over a period of 30 years, beautiful work on problems of synchronization and connectivity. And the question in the chat was on homework 3, are we supposed to have K forward and K reverse constants for reversible reactions? And the latter problem only called for forward ones. And then you said, if I'm understanding the material correctly, the forward and backward constants can just be replaced with a single constant. And I guess somebody would have to actually screen share it for me to be able to answer that question. If it's possible there was an ambiguity in the problem statement. Although I should say that that problem was given 3 years in a row with nobody asking that question. Which is not to say that they shouldn't have asked the question. I just don't remember. I don't remember the ambiguity. In general, we assume that the forward and backward rate constants are different. That there is a k forward and a k backward that are different. Now, if you obviously when if you make them the same, you may get some cancellations of things that make things simpler. Okay, let's see.
So I'm not sharing, Joel, if you'll scroll down to like 3C here, the test the 3 cases towards the end of C. You call out K forward ES significantly greater than K forward EI. The very last one there, you have K forward E. What which of the rules are you referring to that is just E? Because there's ES, there's EI, and then there's E plus P. I wouldn't, in my initial pass through, I didn't define any of my constants as KFE. So I was pretty confused as to which one this was.
Okay, so let's look back at the diagram. So the only possibility for K forward E would be for E turning into ES. But that should depend on the amount of S that's present. So let's see.
Yeah, I guess I'd, like the first problem, even like the hint that you give, it seems your convention is to put both the reactant and the product in the forward, but then down here, if what you're saying, you expected that one to be just E, it's just confusing as to the notation.
Okay, so yeah, I guess I skipped some of this because it seemed sort of arid last week, or 2 weeks ago. So yes, so really what we should write is E plus S goes to complex ES. And we would expect the rate for that to be K sub ES times the concentration to be times the concentration of S, right?
Yep, I'm following.
Right, that would be the first one there, K1 times E times S, yes. And so, but if we assume that if we make S a boundary species, which means we're assuming that the amount of S is constant, then we can combine we can look at K1 times S as a rate constant, because if S isn't changing, K1 times S is always the same. And then K1 times S is the rate at which we're synthesizing ES from E. And so I'm pretty sure that the intent was I'm pretty sure that the intent when I wrote that problem, and I think this is in Herbert's textbook, isn't it? The intent when I wrote that problem was that KE would be equal to KES times S. KE forward. Because forward, if it says forward, it has to be something which has an E on the left-hand side. And I apologize if I set a trap for people. My guess is that what happened was I skipped the slide. I may have skipped a slide should be in the slide deck, but I may have actually not presented a slide that talked about this interpretation. And I managed to forget that was in the homework in that form. But that's why that $sign, a KSE times $S turns into K forward E with no $S. That's what the idea was.
Okay, so if Joel, if you're still presenting, if you would go to C. What you're saying then is K forward E is the first problem. K forward ES is then the ES to E and P. Then K forward EI.
But you're right. Then no, maybe I'm wrong, because you're right that if I have K forward ES there, and I've defined K forward E as being much bigger than it, that would be equivalent to simply saying that the amount of S is large. And so yes, so maybe, boy. That's the three cases.
I thought you were comparing KFES with KFESI with that one, but I guess it's just from whether you take off of. I thought so too, right? The first one is PI, and then the second one is the third one is E.
Right. Yeah, so the first one makes sense, because the question is, do you bind to the, does the enzyme bind to the substrate or the inhibitor more effectively? The problem with the second and the third one is, does the substrate bind to the enzyme? So E, okay, let's see, where's KFES defined? Because that's the problem here. So KFES, that's your definition, or was that something that I gave you?
That's not what you gave.
Yeah. Right. Right. And so, yeah, there definitely is an issue with. Now, if we scroll up. Now, is this the comment E arrow ES means DES DT equals KSE times $S. Well, let me look. Let me see if I did a slide on this. I think I may have skipped. Let me see if I have the slide. Sorry about that, but if you're having trouble, it's probably better that we go through this, because I don't want to. And I'm not going to promise I'm going to figure this out today. But yeah, I apologize if I created a mine for you. What's missing is there's no. Yeah. I'm just looking at the,
this one, I think it would be something like 9.
Well, this certainly illustrates the danger of writing the complex as the two symbols concatenated to each other, doesn't it? So let me see if I just screen share. Let me screen share. Sorry, Sean. So Joel was saying that he was trying. Sorry, I should have put Joel on as the mic. He was talking about his interpretation of the symbols, and okay. So let's see what we have here. We have KF.
That's pretty funny.
KF times E times S, KR times E times S. But here it's not obvious why KF and KF for S and KF for I should be the same. You think that there should really be a subscript for those to indicate which is which. Yeah, so here I've defined KS prime to be KS times S. That's what I was talking about. Here. Here we have KES, so that one is well defined. Here we have KEI defined. And we've got KI prime. So I think the KI prime is probably when I wrote K forward E and K forward, but that's K forward should, if it's I prime, it should be KI, not KE. Yeah. And so I have KS prime, KES, KI prime, KEI. And now let's come back to the homework assignment. If you share it again, yeah. Right. Right. ES, so ES is well behaved, is well defined. KS is well behaved. Yeah. Yeah, I think that's right. And there, yeah. Okay. Because we can't call that K sub ES because then it would be the same symbol as the other one. Yeah. So I think that's right.
Yeah, then it would be the same as the top one.
Yeah, there we go. And so it's actually with the backward, it's actually it's the forward reaction for the complex is KE.
Okay, this is how I did the assignment, so that works.
Well, you're ahead of me. Somewhere I actually have this. Somewhere I have the solutions to that, but I'd have to dig them. I hadn't dug them out yet. I didn't have a chance to grade the homework yet, and so I didn't have them dug out. And I apologize for having set you a trap. That wasn't my intent. I made the point. I unintentionally made the point that the notation is confusing more severely than I intended. So I do appreciate this, and I do appreciate all the thought that was put into it. I'm quite content for you guys to say, hey, wait a minute, the homework problem makes no sense. if it, so think about it for a bit. But if it really makes no sense, tell me it makes no sense. How did other people interpret that, or did people just give up?
We're still in confused state, Professor.
I should say that this problem was a little bit. It was a problem coming from Herbert, and maybe it was a bad choice of a problem to do. The reason, and it's not something that we're going to need within the context of Herbert's textbook, because Herbert's actually quite careful to always write everything out. He doesn't use these shorthands. And so the purpose of this was really to help people read additional, to read the literature. And I didn't mean, it's not worth spending a lot of time trying to figure this one out. And that was a mistake on my part. So at least I can promise this, which is next year I won't assign that problem. But I think that you're right. It's the K forward ES. But the question then is K forward ES and K forward. If ES goes to E plus P, that could be K forward ES. But if EI goes to something, what's the notation for that, right? Yeah, if you use reverse, that makes sense. So KFES and KFEI is basically a question of does E bind to S more strongly or to I more strongly? Does the enzyme bind to the substrate or the inhibitor more strongly? And that is something that happens biologically. Sometimes you have a strong affinity for one, and sometimes you have a strong affinity for the other. So that first test case makes sense. The second one, KFES equals KF. I think that these are all simple. I think that there's a much simpler answer here than this. I think that the correct syntax should be it should be KFEI for all three. It's KFES much greater than KFEI, KFES equals KFEI, and KFES equal much less than KFEI. So EI we defined, which is that one. Yeah. So did I then have the same? Let's look at the question then.
Yeah, this is what the most people I interpreted it as ES to ESI.
Right, so what we want is the affinity of E. Okay, so let's go scroll up. Yeah, this really was a mess, wasn't it? Boy. Let's scroll down. So the ESTT. So we have example of KSE. KSE. No, because E is really a variable, so it's really KS. Okay, so KSE, and now the irreversible one there is your answer. Is that right, Joel?
Yeah,
so, and the issue here is that because I is written as a boundary species, it doesn't show up as it doesn't show up in the name. That's why. And so then, yeah, so then KE K forward ES, there'd be two of them. One is ES going to E plus P, and the other one is ES plus I going to ESI. And the question is, what are those named? And what I was trying to do, again, if we scroll up, the intent of this was to ask the question, does E bind to S more strongly than, in this case, ES binds to I? And so if we wrote E plus S goes to ES at a rate KES, and ES plus I goes to ESI at a rate KESI, those would be the ones we would compare. Right. Yeah, so there, yeah, so if you wanted to disambiguate that, I would write, yeah, there you go. And then we need the I's, the SI, yeah, there you go, and then SI. That would be it. Because again, the purpose of this exercise, boy, well, you've saved me. I've got a lecture in the can for next week this week. But the purpose here was to say there are three cases, one of which you have a lot more substrate than inhibitor, one of which you have equal amounts of substrate and inhibitor, one of which you have more inhibitor than substrate. And then the second case is, suppose you have an equal amount of substrate and inhibitor, but the binding of substrate is strong compared to binding of inhibitor. And so fundamentally, if you have a lot more substrate than inhibitor, but inhibitor binds much more strongly, then you should get the same result as if they were equal. And the binding was the same. So this was actually brought up in one of the examples, right? With the EGF binding to EGFR. We could increase the signal by increasing the affinity between EGF and EGFR. That would increase the signal for the same number of EGFs and EGFR. If the affinity is bigger, we get a bigger signal. For the same affinity and the same amount of EGFR, we get a bigger signal if we have more EGF. If we have the same, whatever the third pair is, what's the third pair? If we have the same level of EGF and the same affinity, and we increase the amount of receptor, we get a stronger signal. So it was basically, suppose it was 3 by 3, because you could say either that you have a lot more receptor, you either have a lot of inhibitor, equal amount of inhibitor and substrate, or a lot of substrate, and you get 3 regimes. Or you could say that you have binding of inhibitor strong, binding inhibitor and substrate's the same, binding of substrate strong. And so in your 3 by 3, basically you're going to get something that's symmetric where they cancel each other out. You're going to get a situation where you effectively have on the off diagonal, you'll basically, things will be balanced. And that was the goal of that homework problem. And I'm sorry that got clobbed. And since we didn't really do that in class. Sorry? Sure, if you like, go ahead. You better turn your mic on so people can hear.
Joel, if you're the one sharing screen, could you turn your mic on, please?
Yep, so here I'm just showing the species. and not the case, because I think on the first question on A, you ask for the species comparison of SE, right? But anyways, just give an idea of how the plots should look like, I guess. That's why I wanted to show, and the between irreversible and reversible, the difference too. But as you can see, the K's are not changed for the last K's. You tell me if you want me to change something.
No.
Okay.
So I mean, I think, again, the concept of the problem, and it probably wasn't worth the effort we wound up putting into it now, was that you have the three regimes for the concentration of substrate inhibitor. And the question was, if you have a lot of substrate and a little bit of inhibitor, what do you have to do to the rate constants to bring it back to behave as if it had equal amount of substrate?
Okay.
And so it was essentially trying to do a global sensitivity analysis without actually telling you what a global sensitivity is.
Okay.
And so if people are thoroughly stuck on that, give me a day or two to try to figure out a better way of framing the problem. If I can, I'll hand it back to you. But I think genuinely, I don't think it's probably worth struggling a lot with that at this point. I think we should. I mean, I can certainly. It's not a hard problem to do, but I don't think it's probably worth the amount of time it would take you to redo it at this point. So I do apologize on the notation that I hadn't realized that got messed up.
Yeah, and I should have reached out.
Anybody else? Were there any other potholes in the homework that we need to discuss?
Somewhat related, I do really appreciate in the fourth problem you specifying the inputs for the parameters when you ask us to define like the shapes of the curves and whatnot.
Sure, I'm happy to let you have more time on it, but before you try it again, let me go over it and make sure that I frame the problem right.
Yeah, definitely.
Okay.
But asking about other.
If I can't reframe the problem in a way that makes sense and is easy to solve, I just will delete the problem.
Yeah. Like that works. But just like speaking generally, I find it difficult to define or describe the shapes of the curves when it's not defined like the parameters, like you specify, to increase the constant forward of ES to be significantly greater than one of these other ones, but you don't define anything about the reverse constants. And in my application, I change those frequently. And so it wouldn't be this nice, it looks the same as one of the previous graphs because I was perturbing many of the values outside of the ones you definitely wanted. So I would find great benefit in you specifying like, start with S equals 1 and only change E here and only change I here, and then move to like different concentrations. But maybe that's just me.
No, I mean that's right. I think it's a sort of a default. I tend to assume that we start with everything equal to 1, unless we say otherwise. But I agree with you that that's not completely obvious. Let me look here. Give me 1 second. I was looking to see if I have that problem done out somewhere. And I'm afraid the answer is no. I do not have that problem done. And I apologize also because I know I did do it. I remember actually solving that problem, doing it out completely. But yes, the assumption I normally work with it, you know, in many cases we know what the rate. I mean, not in many cases. Sometimes we know what the rate constants are. And when we know what the rate constants are, that's great. When we don't know what the rate constants are, typically our workflow, and this is certainly not something we've had a chance to talk about yet. It's something I was hoping to talk about later. Our workflow would be to start with everything, all the constants equal to 1. And then ask the question, if we change one value or a pair of values in some kind of ratiommetric way, what happens? And then the goal here was we say we change one pair of constants in a ratiommetric way, namely the initial amount of E and I, S and I, and see what happens. And now we change one other pair, which is this rate constant pair, and see whether we can recover the initial one. And I agree, we did not cover that. So that was my bad. And unfortunately, it's not the first time in my life where I've assigned a problem that seemed to me to be very innocent, and it was not so innocent. And one of the reasons I'll give you a rough indication of the amount of time that a problem is supposed to take is because that way, if you work on it and it seems to be, wait, gee, he said it should take me 20 minutes, it's taking me an hour. It may well be that I made a mistake. So I don't know what I can do. I may not be able to get to fixing those homework problems until later in the week, though. So if you came up with some answer that you're happy with, submit it. If you didn't come up with an answer, don't submit it. And I won't grade you on that homework problem. If people are frustrated and want to do the homework problem with a cleaned up version, then that's an assignment to me to fix the problem, which I probably should do for next year anyway. But that basically is a hangover from when this course was more mathematical biology. And so occasionally there are going to be places where there was material that got cut and something depends on something. There was a dependency that I didn't realize existed. And that seems to be one of those cases. All right. I guess I would I mean, I'm happy. I don't normally take a vote on something like this, but why don't we? I mean, if people really want to do more homework problems about enzyme substrates interaction, I'm happy to spend time working up these problems to make them more sophisticated and clearer. If people have banged their head on that problem enough and are comfortable with my answer, which is, oops, I made a mistake, and you don't want to be graded on those problems, I mean, let me do it a different way. How about this? I will say that problem set, those two problems are extra credit. We reduce the number of points in the problem set from 40 or whatever it was to 20. And if people handed something in, you'll get a few extra points. How's that? And if you didn't hand anything in, you'll still be scored on what was done. Is that okay? I mean, I don't want to be unfair to people, but I also don't think people have enunciated such great projects. I want to minimize the amount of time I'm taking away from the projects. And I think the learning value of that homework assignment is not great. And I know, thinking about it, I know where that homework assignment comes from. It comes from a math bio chapter. It's in Britain, essential mathematical biology, where he's talking about the kinetics of competitive inhibition. And it wasn't originally written as a computational assignment. It was originally written as an analytic assignment where you find equilibrium values analytically. And so I think it got messed up when I tried to convert it from find the steady states for this, and this to say simulated. I messed it up. But I am a little bit surprised because I know I gave that homework. I didn't use that. It's as they say, an explanation, not an excuse. But this course is a rollback to 2019. Because in 2020 and 2021, I taught a totally different version of this course with totally different material. Covered antimony and tellurium, but everything was in the context of infection and infectious disease. And so I'm reverting to the old class, which I taught multiple times, but occasionally there'll be something like this where it looked okay to me, but I didn't redo the problem. And so and because I was traveling, if somebody yelped about it, I might have missed it, and I apologize. So sorry about that. All I can say is I'll try. I can remember how cross I was with my professors when those kind of things happen. Boy, let me say it this way. I appreciate you being patient. All right. So, boy, let's see, 6:40. What can we talk about then for the rest of the class? Well, we're certainly not going to get through that lecture today. Okay, so just a couple of things we talked about last time, and we talked about this concept of stoichiometric coefficients. We did some analytics on it. Let me get back to that screen share. We did some analytics on it. We talked about reversible and irreversible rate laws. And there were some questions about, do you assume that the forward and backward rates for the Ks are equal or not? And again, my recommendation always, and Joel, that I've worked with on this a little bit more, is that to begin with, in the absence of information, assume all your rate constants are 1 and see what happens. And then ask the question, if you make one rate constant different, what happens? And perturb it up and down. So you're basically doing a sensitivity analysis for yourself. One of the things that you do find, though, sometimes, and that's a homework problem that I think we may get to next week is even in a very simple cascade reaction. We did that a little bit in class. A goes to B, goes to C, goes to D, the forward reaction. If all the K's are equal, K1 equals K2, K equals say 3, you get a particular behavior. If you have K1 equal to alpha, K2 equal to alpha, K2 equal to alpha, K3. So you have a geometric series where K1 is equal to alpha, K2 is equal to alpha squared, K3 is equal to alpha cubed, K4 is equal to alpha 4. And so you have a geometric series of rate constants. Then you can get some interesting behaviors as you change the ratio. And so in those situations, just changing one value at a time doesn't do it, because what you have to change is basically the whole relationship of the stack. But a lot of times we're going to face that we don't know what the rates are. They haven't been measured experimentally. And your best bet is to assume that they're equal to begin. And in some of Herbert's textbooks, he'll give examples where you'll see very funny, you'll see values of 147 and 0.01 and things like this. Typically, when you're going to do that, the way you get those is by going backwards. You say, we have a chemical species, how long does it last? This molecule is pretty stable, it sticks around for days, so its decay constant has to be 1 over days. This molecule disappears pretty fast, so its rate constant has to be 1 over 10 minutes. So in those kind of situations, you may get different values because you have a concept of how fast things occur. In the case of binding affinities, you depend on concentration as well as rate, and that makes it a little bit harder because you don't know how long something takes unless you know the concentrations as well. And so typically we're also going to scale concentrations to be between 0 and 1. We'll say the amount of A, the concentration of A is 90% of whatever its typical maximum is. And that's a slide deck that I have coming up in a week or two, which is if you have a network diagram and you don't know anything, how can you make some sense out of it to start working with it mathematically? So we talked a little bit about irreversible rate laws and reversible rate laws. And here you'll notice, for example, we have VF and VR here as the forward and backward rates. If the parameter values were equal, those cancel. And so here we've expanded the V's to be KF A cubed, KR B squared. I didn't use the subscript A and B here, which is probably not a bad thing. And we talked a little bit about the boundary species idea, which is what's been confusing us here. And that issue of notation, mass action. So coming back to this idea of making KF and KR1 to start with, making KF and KR1 only makes sense if our concentrations are also scaled to be between 0 and 1. Because if the concentration of A is between 0 and 1,000, and the concentration of P is between 0 and 1, making KF and KR both of order 1 will give vastly different rates on the two sides. And so that really is the problem. And we derived the the steady states and their dependence and their chemical equilibria, and we analyzed their dependence on the parameters. And that, I think, was probably the main result last week. That this concept of a steady state and the concept of an equilibrium seems very similar, but we get rather different results in the sense that in one case, A star, the steady state, doesn't depend on the initial amount of A and B at all. In the other case, the equilibrium amount of A and the equilibrium amount of B depend on the initial amounts of A and B. In one case, the ratio of the steady state amount of A and the steady state amount of B versus the equilibrium amount of A and the equilibrium amount of B. Notice in this case, it's more similar. A star over B star is KR plus K2 over KF. In one case, it's just KR over KF. K2 is the decay rate. So if the decay rate is small, the equilibrium ratio and the steady state ratio are almost identical. And of course, the steady state one is just K2 equals 0. So they become identical. So that was something that was sort of important. This was the beginning. See, this is beginning to generate that homework problem, right? We had E plus S goes to the complex ES and then decays to E plus P. And what was different was we added a side branch. So this was our basic reaction. And then we added a side branch, which was an inhibitor. And what we were doing is we could add that in two places. We could either take E out of circulation on the left-hand side of this, or we could take ES out of circulation. And the question was, if the inhibitor bound to the inhibitor bound to S, how did it behave? If the inhibitor bound to ES, how did it behave? How are they different? And that was really the point of that homework problem. Although that wasn't really enunciated. And the last thing we did was just at the very end was this issue of how to display things in Tellurium and selecting column outputs. And so I don't know whether it's worth doing this or not at this point. Did people have the code we were doing at the end of last class available to them? Do people remember that? Well, we could do that, yeah. Because the homework problem, that what I was going to do here was just do some quick plotting examples. And actually, this Michaelis-Menten enzymatic reaction V2B should be in the problems for the. It should be available to you in the demos. Yes, it's there. Is it uploaded? No. Why not? Great. All right. Let me get it. It looks sorry, it looks like that one didn't upload. It was supposed to. Okay. So let's start from where we were. Joel has a version of it. Here's my version. And why don't we all try to get that? So I put it in the chat so you don't have to upload. Make sure it works for me. It does. Okay, so let's come back and see if this works for people. Was everybody able to grab that code from the chat and put it into NanoHub? We don't have very much time, and I got to say I was derailed is unfair. It was more important to try to figure out what was wrong with the homework problem, and it was more important to hear what people were thinking than to go through these examples. But I have been trying to mentally figure out what would be reasonable to do for 15 minutes, and that's been a little bit of a derail. We're clearly not going to do anything very profound in 15 minutes. I figure we can play around with this a little bit, just in terms of using to learn. So if people can get this, does everybody have this working? I'll wait until make sure everybody's. Somebody says it works, but so the main point here is just to use this list to select the things that you want to output. Because I introduced that as a concept at the end of last class, but we didn't actually work with it. And I don't know what this PyLab thing is. I don't know why that's. Actually, I do know why it's there. It's there because this was originally done in Spider. Does anybody, so how are people, how is it coming? So people need, Grant, you got it working. Does anybody need more time? Has everybody got it? If you need more time, raise a hand. Okay, everybody in here all right with it? So, okay. The main thing that I wanted to do here was to say that one of the things that you can do, which is useful, is that a lot of times what you want is not the value of the species, but the value of the rate count, the rate law. And so if I look here in line 4, I've named this rate law J1. And by default, if I just do an R dot simulate, I don't get J1, J2, and J3 returned to me. But if I want J1, I can type it here. So I can get J1 and J2, and I can tell it to give me the grades. So why don't you try that? Just use the names, the named rate to get the rate law. And that's reasonably convenient to have. Again, it's nothing tremendously sophisticated, but I just want to make sure everybody got some. By the way, if I didn't have time here, what would happen? I don't have time. It should treat J1 as if it were time. And it should plot the value of J1 against J2. It may just crash because it may insist that it find time. Let's see. No, it plots J1 against J2. So the R dot plot, whatever the first column is the x-axis, whatever the second column is the y. If people get that. So if I want to know what the rate of production of P is, that's J3. So let me plot J3. That's the rate. Okay. So this is actually now telling us something a little bit. We've sort of forgotten about what this enzymatic reaction was telling us probably from last week. But this is now telling us something which is actually significant. And notice that the rate constants here are all equal to 1. Kf, Kr, R are all equal to 1. What it's telling us, it's a little bit, that's reasonably significant, is that with the rate constants all equal to 1, the rate at which we produce product from substrate through this enzymatic reaction increases rapidly. Within a half a time unit, our rate of reaction is maximal. Right? Our rate of reaction happens of formation of complexes very fast. And then we start producing P, and the rate of production of P goes down, but it goes down very slowly. It goes down from 0.8 to 0.7 over 5 time units. Yeah, go ahead. Right, well, if I make the time bigger so that I run out of substrate, then of course the reaction will turn off. So let's try that. Make the time 25. Well, if I run out of substrate, the reaction turns off. But what this is trying to investigate is what's called the quasi-steady-state approximation, where you assume that the amount of substrate is big enough that you're not using it up. And there are plenty of reactions where the thing that's being reacted upon, there's a lot of it. And in that case, we're not forcing it to be constant. If we put a $ in front of S, then it would be saying we're assuming there's an infinite amount of substrate. In this case, we're not doing that, but we're saying over a short amount of time, there might as well be an infinite amount of substrate. And so in this case, that enzymatic reaction over periods of a certain amount of time is behaving as if the substrate concentration were constant. And one of the things that you could do that would be interesting here is exactly what Joel talked about, which is ask, for how long is my assumption that the rate of production is roughly constant reasonable? It's going to depend on how much tolerance I have for variation of that rate. But if I want it to stay within 10% of its initial value, then I'd say, in this case, over 3 time units, the rate of production is approximately constant. If I increase the amount of substrate to 100, you'll notice over 10 time units, effectively, it's absolutely flat. But of course, if I stretch this out long enough, it'll still go down. And something interesting here is I increase the amount of substrate, the rate of production of product stays pretty constant out to 190 time units, and then it decreases very rapidly. And that's something also that's an interesting behavior. So even this very simple enzymatic reaction has some temporal behaviors that are interesting. The equilibrium values are simple, but the temporal behaviors are going to depend on the ratios of these parameters, and particularly the amount of substrate compared to the amount of enzyme. And here we could do what we talked about, which is we could now fix the amount of substrate. And now, of course, it really is a straight line. And you can then ask the question, how different are they? And the answer is, how different they are depends on, first, what level of difference divergence you're going to accept, 5%, 10%. Secondly, how long you run it for. And thirdly, how much substrate you start with. And so this was the one that I would like you to do in our last 5 minutes, which is let's do the same simulation now, and let's do it twice. And you could do this in a couple of different ways. I'll let you solve it the way you want. In one case, solve it with the substrate limited, and the other case, solve it with the substrate not limited. Ask the question, how much did they diverge? Does that make sense? Right. In one case, put a $sign in line 4, in the other case, don't. And so you could do this in a variety of ways. I'm going to do it just in a dumb way by cutting and pasting. This one will have the $sign, the second one won't. That may or may not work. Let's see. So put them together. Oh. That plot is not. The way I did it here, I actually put them in the same plot. It works, it looks nicer. Then the colors. Does anybody get that to work in to get it prettier or make it more look more like what I had there? Yeah, the way I did it here, the way I did it in this one was this. As I said, they never thought there was going to be more than one cell in them. Not very pretty. Oops. Named array. Oh, please. Named array has no attribute flaw. But today. Oh, because it's operating on not only it's not operating on result. There we go. Except here, I don't want to be limiting my. Yeah. I realized that seeing me code isn't very edifying, but I was hoping that people would be playing with it on their own. Yeah. I think it's a question of if you wanted to give you the color labels, then you have to do a little bit more work if you do it with this one. Question? Yeah. Oh, so I was doing something which was not very elegant, which is I want to run the same model twice. Once with once with S fixed and once without it fixed. And I can do that in two ways. I could copy my model and have two independent models, one of which has S with a $sign and one of which doesn't. Or in this case, since all the parameter values are the same, I just cloned the model definition inside of one model string. But I can't have the names of the variables be the same. I can't have E for both. So I just added an A to the end of each variable name. This way, if I change the parameter value for one of them, I automatically get the parameter value changing for both. And something that I didn't say, which I can do here, is that I can actually do a successive assignment like this. If I want to make sure that S and SA are the same, no, it won't work. Doesn't like that anymore. No, won't let me do that. Right. I'm wrong. They used to let me do that. They changed this in. All right. What did I do wrong? Oh. So now I can compare the result of fixing that substrate value. So the production of P, the amount of P I get changes. It might be more interesting, instead of comparing the amount of P, to compare the rate of production of P, which would be J3 and J3A. And here it's pretty clear. In one case, it's constant. In the other case, it decays. If I start out with more substrate, they're identical for 10 time units. If I go to 50 time units, they're still almost identical. So I know that was a bit rushed. I don't want to keep people over. We've been patient to them. But this actually is a more interesting problem to work on than the one I gave you as a homework problem. And so I thank you for being patient. I will sit down with Joel and we will get that homework problem fixed. Not that I am assuming people will do the homework problem, so that for my own sake, I want to make sure that at least I don't have that trap like the people next year. I really appreciate the neat projects that people are coming up with. And I exceptionally appreciate the fact that everybody was ready to present your project when I was only expecting you to present what you were supposed to have done for last week. So everybody's actually a week ahead of where I thought you'd be. And so I would like to try to meet with groups. It may take me a day or two to catch up, having been away. There's a sort of a stack of unanswered emails, as you know. But I would like to try to meet with people in the next couple of days to talk about projects, look at the papers, your source papers, and to try to help you begin to refine the ideas. But as I say, everybody has fantastic topics. I just want to help you make sure that you narrow them to something that's productive. So any comments or questions before we break for the evening?
So is there anything due next Tuesday that is for the project?
Let's see. So I think everybody actually did the presentation of their project idea today, which was not what I planned. I planned on that next week. So everybody's ahead of the curve on that. I think that for many of these projects, there's still, as I say, you've got great topics. Turning them into a workable project is going to be work. And so I would encourage you to start, to spend as much effort as you can on refining those into something that's manageable. And as I say, I would like to meet with people, try to give each group an hour of time to discuss. Now, it looks like there were about 7 groups. And so my finding 7 hours between now and Friday is problematic. I can probably find 2 or 3 hours between now and Friday, but that doesn't get everybody in. I am happy to meet with people on weekends. So if you guys are available to meet on a Friday, a Saturday afternoon or Sunday afternoon for an hour or two, those usually are easier because then I don't have other administrative responsibilities that time me up. I won't insist that you meet on weekends. You're allowed to have free time. But if you're available on weekends, I'm always there. And it does have the advantage that I'm much more flexible in my schedule.
I know, at least to speak for my group, since our partnership is pretty fresh, we probably wouldn't be ready to meet until sometime next week anyway, so we can actually like dive more concretely into the connecting literature and make sure we understand what we're getting our hands dirty with.
That's fine. I don't want to force people to meet, but I do think, again, my worry is if people pick something that won't work. And just as this homework problem, there could be a trap in an apparently innocuous homework problem that I miss. So I won't guarantee that I will necessarily catch something where you have an idea, it looks like it should work, and then it doesn't. It can happen. But I've done this enough that I have pretty good sense of what kinds of problems won't work. And so if I can try to help you avoid that as early as possible to minimize the kind of time wasting that you did on that homework problem, I want to be available to do that. And so if you need more time and you don't want to talk till next week, that's fine. But I want to make myself available as much as possible, as soon as possible, especially since I was out of town last week. So I was teaching Computational last week. Trying to get a semester course into 3 hours. That was a challenge. Okay, well, thanks, everybody. I appreciate you being patient, and thank you for catching mistakes. I have to give some extra points to everybody who caught the problems with that homework problem. I promised everybody would get some extra credit if you identified mistakes in the homework. So I will definitely give you some extra points for that. And next time we'll talk about Michaelis-Menten forms and Hill forms. And given the level of the class, I probably will push through that pretty fast and try to get to do some motif analysis, which should be more interesting to people. Okay, good night, everyone. Thank you.
Thank you.
Thank you. Bye-bye.